Okay, so I will talk about the secret life of soils, or actually it's more of a sort of a personal journey into the secret life of soils. But at the same time, it's also an ode to all the people who inspired me to work with soils, because really, I never intended to study soils at all. This all just happened. So in 1996, I started to study environmental sciences at Wageningen University, and really all I wanted to do was to be a scientist on a Greenpeace boat. So I wanted to go to the Arctic, take water samples and analyze them in those boats. This just seemed really exciting to me. But in my third year at Wageningen, actually these two people, Leibert Brussaert and Ron de Goede, who at the time looked a lot younger, and I can say this because they're not in the audience, they inspired me with their lectures of island biogeography and ecology of soil animals. And this was mostly work in their early days on ground beetles. So I decided I wanted to do my master's thesis with ground beetles. So I went to see Ron de Goede, and I said I want to work with ground beetles. And he said, well, we don't really work on ground beetles anymore, but you can study earthworms. So I thought, well, okay, not quite the same. And I was going to study earthworms underneath cow flats. But then the foot and mouth disease took off, and there was just no way that I was going to go into grasslands and look at cow flats, because it was just a no-go area. So Ron actually suggested that instead of earthworms, I looked at nematodes. So even smaller animals, well, you can't really even see them with the bare eye anymore. But okay, I did work with nematodes, and I looked at the effects of nematode diversity on nitrogen mineralization. And I found that they're actually quite important for nitrogen mineralization. So this all went really well, and then Alice Hofland sent me this advert about a PhD on fungal bacterial ratios, which Richard talked about this morning. So, and I actually wrote in my application letter that I wanted to make agriculture more sustainable. So I got the PhD, and there I was, looking even deeper into the soil at organisms that you really can't see with the bare eye anymore. So I spent hours and hours looking through the microscope, and as you see, I haven't aged at all, looking at fungal hyphae, counting fungal hyphae and bacterial cells. And one of the things I did was a field experiment, and I looked at the relationship between fungal bacterial ratios and nitrogen leaching. And I found that the more fungi you have in your soil, the lower the amount of nitrogen leach. So this was in a field experiment at Aveheino in Overijssel. So after studying fungal bacterial ratios for 4 years, really there was no option but to work with Richard Bargett. Because basically my entire PhD was based on this iconic paper that he wrote in 1999. So I went to do a postdoc with Richard in Lancaster in the UK, and I couldn't really let go of the fungal bacterial ratios. So I wanted to see whether this was a general relationship. So I looked at 60 grassland soils, and I found that when you have a higher fungal to bacterial, that you have lower nitrogen losses as leaching. So, but grasslands weren't the only systems I worked at. I also looked at more intensively managed agricultural systems, and I found that when you subject these soils to drought, that when you have more fungi in your soil, the soil food web is more resistant to the effects of drought, but it also retains the nitrogen better. But if we talk about soil organisms, really, we are only talking about half of the story, because plants and soil organisms are tightly linked, and this is an iconic picture, and this just shows you that plants, through their plant traits, which determine the quantity and quality of litter, have a tight link to soil organisms. So I went back to grasslands, and I found that plants with slow-growing, conservative plant traits, leaf traits, high dry matter content, on a landscape scale were linked to more fungal-dominated microbial communities. But really, looking at leaf traits doesn't make sense when you want to look at microbial communities, because it's the roots that are in direct contact with the microbes. So these root systems, they differ immensely between species, and these might actually be the key to selecting beneficial fungal-dominated microbial communities. However, in agriculture, we've selected for fast-growing plants with really measly small root systems, such as this wheat plant. So these doesn't actually seem very beneficial when you want roots to harness soil microbial communities. So what now if we could actually make use of this huge variety of root systems in agriculture and select root systems or put them together in a field, such as intercropping, and use those root systems to actually select for microbial communities that we want in the soil. So there's several ways through which we can also do that. Besides, for example, intercropping, we can also, for example, use perennial crops rather than annual crops, because the roots stay in the soil and they're not destroyed and plowed, and the microbes' root linkages remain intact. So this is an option. But also, one other possibility is to actually select beneficial root traits, for example, through crop breeding, and to select beneficial root traits that have close linkages through root exudates and root architecture with microbial communities that have beneficial effects for the plant growth and resistance to drought, for example. So in the end, I am really very pleased that I didn't end up on a Greenpeace boat and that I didn't end up studying ground beetles, because really I think the key to a more sustainable agriculture is in the soil. And also my soil work has brought me to the most amazing places like this glacier in Austria. Thank you.